Welcome to this, the fifth lecture in our Chemistry of the Kitchen series. We'll be discussing seeds in this lecture, in particular the cereal grains, and we'll talk about the chemical composition of cereal grains and the importance of carbohydrates and proteins in bread making and baking in general. So we're going to begin in Chapter 9, and we'll wind up somewhere in Chapter 10 of your textbook. Starting though with this rather complicated drawing of a seed, let's think about the key parts of the seed. First of all, of course, there's the germ. That's the part of the seed that's ready to become a new organism, the embryo. The seed is protected with this exterior coat known as the bran. This is most of the pigmentation is found in the bran. Lots of the vitamins and fiber is found in the bran. Most important for us, though, is going to be the endosperm, because the bran and the germ are generally swept away when we mill and refine our grains, and what we're left is an endosperm that's largely starch and protein. Let's look at the classes of seeds. We're not going to discuss in this lecture the legumes or the nuts, which are described in Chapter 9. We'll come back to those. The legumes, also known as the Fabaceae, domesticated around 10,000 years ago, they'll be very interesting to us as a source of nitrogen. So they have a symbiotic relationship with bacteria, a certain group of bacteria that are involved in fixation of nitrogen and enriching of the soil, therefore. Quite a long list of cultivated beans. This is just a nice selection of some of my favorites. Some interesting ones here are lupins. Some cultures actually do cultivate lupins, which we're familiar of as flowers that we see around here in the springtime. Nuts, a much broader set of organisms than either the legumes or the cereals, storing their energy as oil, more than 50% by weight for most nut seeds, as compared to the cereals, in which the oil content never really exceeds 3%. But what we're really interested in today are the cereals, the Poaceae, grasses that have been selected for domestication because of the characteristics of their seeds, particularly the fact that the seeds are edible. The era corresponds to that of the legumes, as well as some of the domesticated animals that we talked about last time. Again, for us, the most important aspect of the cereal is going to be that endosperm, the starchy part of the grain that supports the development of the germ. This is just a beautiful picture of wheat. Most of us are familiar with cereals as something we eat in the morning, the so-called breakfast cereals, that are really a processed food that emerged in the 19th century as some interesting food crazes developed. Some of the people that were prominent in those movements, Kellogg, Post, those are names that are still familiar to us today. Typically, these cereals are heavily fortified with vitamins. This is a theme that we'll come back to and have seen already, in fact, with dairy products. Modification of our foods to fit dietary requirements that aren't being met. The other thing I want to say here about the cereals is that they're an incredibly important part of the world food supply. While I'm showing you wheat here, because that's mostly what we're going to focus on today, wheat is not the most grown crop in the world today. It ranks number 3 behind corn and rice. Corn, almost 800 million tons per year. Rice, around 650. Wheat, more like 600 million tons of grain grown each year. This is a more or less exhaustive list of the domesticated cereal grains. Of course, the wheats are a very large number of species that are cultivated. subspecies varieties, cultivars that are cultivated around the world. We won't get into the breadth of that diversity. Really, what I want to show you here is the scientific names of these are interesting to know. Zea maize for corn, Oryza sativa for rice, the triticums for the wheats, Cicalis for the rye, Triticale, a combination of the triticum wheat and the Cicalis rye. Barley is going to be a big one for us, Hordeum vulgare, oats. Those are the familiar ones to us probably. You might have heard of sorghum and millet as well, but really those first top 5 or 6 are what we in the West are mostly familiar with. And we see these used principally as food sources, also very important in fermentation of alcoholic beverages, also as fodder for fodder for livestock. Corn in particular has become important as a source of biofuels, although it's been in recent years supplanted by non-food product-based biofuels. And there are industrial applications such as plastics for maize as well. The other reason to show you this slide is to point out that there are some interesting, more localized grains that are grown around the world that have characteristics that are particularly useful in their environment. For example, fonio and teff, those are 2 grasses that are cultivated in northeastern Africa, Ethiopia, and the Sudan. Fonio is particularly tolerant to drought conditions, poor soil conditions. It grows very rapidly, 6 weeks or less from planting to harvesting. That can be very important for certain kinds of cultures. Teff, on the other hand, is tolerant to a wide range of conditions, ranging from drought all the way to flood. The zizanias are also interesting, a very relatively small number of species cultivated around the world, most of those in the New World, North America, one species, as we'll see, grown in Asia. A pretty minor source of cereal grains compared to the rest. Sorghum is worth mentioning as well, principally used as a fodder these days, although I'm sure there are plenty of human consumption uses as well. Millet is worth mentioning as well, again as a drought-tolerant species, but the most interesting one perhaps on this list is quinoa. Not a grass at all, in fact, not from the Poaceae, but a member of the Amaranthaceae, amaranth family. This is an interesting one, domesticated in the Andes in South America, tolerant to high altitude, unlike most of the other grains that we're talking about. Slow growing, of course, high altitude, low temperature, but also it provides a complete protein. That's a term that we hear in the nutrition circles a lot, and all that really means is that unlike the other grains, quinoa provides all 8 essential amino acids. That is, the complement of amino acids that we are unable to synthesize in our own bodies. If you're curious to remember what those are, I refer you back to Lecture 1, in which I gave you an exhaustive list of the 20 amino acids, as well as highlighting which ones were essential. Let's take a look at the geographic development of grass domestication. Certainly, as is probably not surprising, most of the grains that are familiar to us emerged in the Fertile Crescent, in the Near East: barley, wheat, rye, oat, sorghum. Again, these are all about the same time, and approximately the same time as we're seeing domestication of those livestock species that we talked about last week. And from the Near East, these grains spread east and west through Europe and Asia as well. And obviously, very important to the food supply today as well. Also worth mentioning are millet and rice. These are domesticated in Asia, and I mentioned that there's one wild rice species that's domesticated in Asia as well. Again, all of these around 8 to 10,000 years ago. Teff and fonio, here I'm just pointing these out to you. Again, domesticated in Northeast Africa. In North America, we see zizania, the wild rices used by North American Aborigines, and maize, also North American, a Mexican domestication, but spread north and south and was used widely by Native American cultures in both continents. Finally, again, quinoa. They're important in Andean cultures. Okay, so I'm going to talk about a couple of grains in particular, just giving a few details. I don't have time in this lecture to go exhaustively through these. I'll come back and touch on some as we proceed in subsequent lectures, but barley is a favorite of mine, so I wanted to give you a little bit of barley background. On the left here we see a photograph of barley, a 2-row and a 6-row variety. I'll come back to what that means in just a moment. First of all, let's look the point that I made previously, the wild barley distribution, these filled circles. Current wild barley distribution correlates very nicely to the location of early Fertile Crescent civilizations, which makes perfect sense, right? These are the places where civilization was emerging. These are the same places where domestication is taking place. Interestingly, wild barley, and some cultivated barleys as well, are called 2-row barleys, and that refers to the 2 rows of seeds that are growing along this seed spike. 2 rows of spicules growing along the seed spike. The 2 rows, as I mentioned, are wild, and the cultivated varieties of 2-row are actually lower in protein and higher in starch than the 6-row varieties, and so we'll come back to these and their importance in fermentation of starch to alcohol in this and later lectures. The 6 rows, again on the right, were selected for agriculture. So this is the main domesticated version of the barleys, and important because what we see is an increase in the number of fruits per seed spike. A little bit more on that topic. Again, wild barley 2-row single spikelet. Now each flower could possibly have 3 fertile spikelets, 3 that could develop into seeds. But in the wild barley, we see that only 1 develops. It's considered that the real reason for this, in terms of adaptation and evolution, is that the single spikelet is more arrow-shaped and more able to find purchase in soil, find its way through other plants, around rocks, and back down to the ground. The 6-row developed, as I said, 8 to 10,000 years ago. This obviously is going to increase the yield per plant, and that's going to be important for a domestic crop that's used as a food source. What we see is that the 2-row was reintroduced to Europe from the Near East around the time of the Crusades. Why would we go back from a 6-row and develop a 2-row domesticated species? Again, high in starch, good for fermentation. The molecular biology of this adaptation is also really interesting. Bear with me if some of this terminology is new to you. When we go about seeking to identify the genetic basis of this switch from 2 to 6-row barley, what we find is that the changes all center on the second or 2H chromosome of barley, and in particular at a particular gene that encodes for a protein called a homeodomain. In this case, it's a rather unique protein that involves a leucine zipper that ties together 2 different homeodomains. The protein encoded by this gene, called VRS1, usually acts to suppress spikelet formation so that we keep the number of fertile spikelets down to 1. That's a 2-row. When we mutate that gene, as happened several times in the development of 6-row barley, we desuppress that spikelet formation, and that allows the development of those fertile spikelets. That's 6-row barley. Rice is another very interesting grain. We've already mentioned it in terms of its role as the second most important grass crop worldwide in terms of the mass of rice that's produced each year. It's interesting because this polishing step that results in the wonderful white grain that we're all familiar with is a result of the similar process that wheat is exposed to in grinding and milling the grain. Removal of the bran and of the germ. This process, of course, winds up depleting the grain of a lot of protein and a lot of essential nutrients, which are concentrated in the bran. In particular, vitamin B1 or thiamine is removed, and this has an important and interesting history in terms of the discovery of vitamin B1 as the first vitamin. Beriberi has been observed in Asia, where rice has always been a very important grain crop, for the last 4,000 years or so, and beriberi is endemic in some parts of Asia. We now know that's to be a thiamine deficiency that arises as the result of a large consumption of rice, in particular polished rice. Well, Japanese naval doctor in the 1880s saw a connection as well amongst crew members that ate a diet that was very rich in polished rice and very little else. He saw the connection and was able to perform an experiment wherein an entire battleship was only given a polished rice diet, and another battleship was given a more complete diet where they had complete protein and all the other vitamins that come along with that diet. And what he saw, of course, was a dramatic increase in the number of beriberi cases in the polished rice battleship, and significantly fewer cases in the more complete diet. This led him to chase down exactly what the essential component that was missing that leads to beriberi, and that was the first vitamin that was identified. We now know that vitamin B1 is important for carbohydrate metabolism. In particular, it's important for the degradation of pyruvate and the introduction of carbon into the citric acid cycle. We won't get deeper into that metabolism today. But it's also interesting for us in terms of yeast fermentation. So B1 is an essential component in the enzyme that converts pyruvate to acetaldehyde, and of course acetaldehyde is then converted to ethanol. So we're going to see this again when we talk about alcohol, but I like to point out the important role of the vitamins in metabolic processes that are relevant to this class. Now, a little more biochemistry. This is called a thiazolium ring, and the position of these two electronegative atoms makes this carbon more acidic, and it loses a proton, and that makes it a really good candidate for nucleophilic attack on, in this case, pyruvate. Good stuff, rice. Let's talk about the evolution of wheat, and in this case I'm really talking about the domestication of wheat. So how many genomes do you have? Well, we all have one genome, of course, but what I'm really getting at here is the numbers of copies of a particular genome. Most higher organisms are so-called diploid, so that they're ploidy, have a ploidy of 2. That includes humans, of course. Triticum monococcum, or einkorn, that's an early domesticated wheat, is also diploid. Durum wheat and many other varieties of wheat are tetraploid. That is to say, they have 4 copies of their genome, as opposed to the normal 2, more standard 2. And then bread wheat, Triticum aestivium, is hexaploid, 6 copies of its genome. Now, that sounds like a lot of genomes, but it's by no means the highest ploidy in the plant kingdom. In fact, it's recently become to be understood that most angiosperms, that is flowering plants, are or have been in their past polyploid. So it seems like this is a common occurrence in plants, in particular in crop plants. Many crop plants tend to be have polyploidy. It is possible that polyploidy could be responsible for traits considered important in crops. So this is a simple answer. More chromosomes means bigger, more fertile, etc. Not necessarily true, but there could be some truth in that. What is clear is that once we duplicate those genomes and we have multiple copies of the genomes, we start to see gene loss from those extra copies. And it is thought that this gene loss that happens after we add more chromosomes gives rise to the diversity that we see amongst closely related cereals. So an increase in the gene density, if you will, in terms of the number of chromosomes, followed by loss of those genes can lead to a wide variety of variation. Let's think for a minute about wheat categories. There's a lot of ways of categorizing wheats. Typically, we'll think of them as either hard or soft, whether they're sown in winter or spring, whether they are red or white. Those are the general categories. Probably the most useful for us is just going to be to think about hard or soft wheats. Hard wheats are wheats that have a high gluten content. Their protein content is higher. In particular, a pair of proteins that are going to be relevant for our discussion that make up the gluten proteins as opposed to other proteins. Soft wheats have a lower gluten content. Wheats can be sown in the spring or the winter, as I said. Seed color can be red or white. More red, I should say. Even white wheats have a reddish or brownish coloration. We'll talk about the origins of that coloration in other lectures, but in general, that coloration is because of a high concentration of polyphenolic compounds. Here's just a list of the protein concentration in these various varieties of wheat, and you see that they range from on the low end to 8 or 9% up to over 15%. Durum wheat is very high in glutens as well, and that makes it one reason that it's so important in noodle making, which we'll see again a little bit later in a video I have to show you. But keep it in mind that the protein content of wheat varies from species to species, and controlling the protein content in our wheat products is very important for controlling what? Our food experience, of course. Let's take a closer look via electron microscopy at starch granules. So we talked about the importance of the proportion of protein relative to starch. What we see when we look closely is that inside the endosperm, these cells are filled with these starch granules. We can see these blobs here as starch granules, and these starch granules are generally embedded in a protein network. If we look at soft wheat, we see that we have starch granules with a relatively dispersed protein matrix, and as opposed to a hard wheat, maybe that's only a 5 or 6% increase in the protein concentration results in this dense matrix of protein. Okay, so here's again our picture of the grain molecule, and we see that there's this